Welcome back, guys! I can't believe it, but we are already on our last topic for Bio 181, and it's a pretty fun one if you ask me. In the previous video, we learned about meiosis and how we obtain genes from our parents. Today, we will go more in depth into genetic inheritance. If you go on to take a genetic class, the two main concepts you should know going in are Punnett squares and pedigrees. Since these topics are really important, I'm going to break them up into two videos so that we can focus on one concept at a time. Before getting into Punnett squares, we first have to talk about the father of genetics himself, Gregor Mendel. Gregor Mendel was known for his experimentation on pea plants, which revolutionized our understanding of how genes are inherited. There were many factors that Mendel experimented on with pea plants. These included pea, pod, and flower color, as well as plant size, pea and pod shape, and flower position. After numerous years of studying pea plants, Mendel created the laws of inheritance. There are three laws of inheritance: the law of dominance, the law of segregation, and the law of independent assortment. The law of dominance was created from the observation that some traits were more common than others. For example, when a pure tall pea plant was crossed with a pure short pea plant, all of the offspring were tall. This means that the gene for tall pea plants is dominant over the gene for short pea plants. Inversely, we could say that the gene for short pea plants is recessive to the tall pea plants. Now, in this example, I stated that both the tall and short pea plants were pure. Another word for pure is homozygous. This means that the tall plant is made up of two alleles that code for the tall gene. These alleles will be represented as two big T's because they are dominant. Similarly, the short plant is made up of two alleles that code for the short gene. These alleles will be shown as two little t's because they are recessive. In this example, we are using t's, but in reality, it doesn't matter what letter you use as long as you use it to represent both dominant and recessive alleles. So we know that when the two plants were bred, all of the offspring plants came out tall. This result would be described as the phenotype. The genotype, however, would be described by the alleles. We can find the genotype by creating a Punnett square. If you haven't done a Punnett square before, get used to it because it might just become your best friend when dealing with genetic inheritance. We begin with a square that has been divided into 4 equal parts. Next, we will assign the two different sized pea plants to the top and side of our Punnett square. In this case, I will put the tall plant on the top and the short plant on the side. Next, I will write big T on the top because it is homozygous dominant, and I'll put little t on the side because it is homozygous recessive. Now, we know that each plant will donate one allele to each genotype. So in the first box, we take the big T from the tall plant and the little t from the short plant, resulting in the genotype big T, little t. I will do that in each square, and once again, they are all big T, little t. So we can say with 100% certainty that all of the offspring of a homozygous dominant and a homozygous recessive trait will create what we call a heterozygous genotype, which means that only the dominant allele will be expressed. In this case, even though all of the offspring had one short allele, the tall allele is the only one that is expressed in the phenotype. Let's take it one step further. The heterozygous offspring we just had self-pollinates. What would the genotypes, phenotypes, and probabilities be for the possible offspring? So we have big T, little t on both the top and side this time. The first box is big T, the second box is big T, little t, the third box is big T, little t, and the last box is little t, little t. The phenotypes would be tall, 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 and one short. If you were asked what the probability was that the offspring would be tall, you would say 75% or 3 to 1. Now, I know this seems pretty easy, but it can get a little tricky later down the line, so make sure you get the basic Punnett square down now. After that long explanation, let's go back to the laws of inheritance. Starting with the law of dominance, which we just learned, a dominant gene will mask a recessive gene, which results in only the dominant gene being expressed in the phenotype, as we just saw with the previous example. Next, we have the law of segregation. This law states that alleles randomly segregate into the offspring. This was shown earlier when the heterozygous plant self-pollinated. Even though there was a 75% chance the offspring would be tall, there was a 25% chance that it could be short. Lastly, we have the law of independent assortment, which states that traits are independently inherited from one another. This means that the genes for plant height are inherited separately from pea color. Mendel was able to prove this with what we call a dihybrid cross, which is essentially a massive Punnett square. As it states in the name, a dihybrid cross refers to the testing of inheritance on two different traits, giving us a 4x4 square instead of a 2x2 square. Let's use the pea plant height example from earlier and now add pea color. We know that tall plants are dominant over short plants. For pea color, yellow colored peas are dominant over green colored peas. Now picture that we have a pea plant that is heterozygous for both height and pea color. Let's attempt to pollinate this pea plant with itself. We have big T, big Y little y on both sides of the dihybrid cross. The first step is to distribute the alleles. To do this, we take the first T and the first Y to make big T, big Y. Then we take the first T and combine it with the second Y to make big T, little y. Then we take the second T and combine it with the first Y to make little t, big Y. And lastly, we take the second T and second Y to make little t, little y. Now we do the exact same thing to the other side. It is important to note that in this example, the x and y axes have the same allele arrangements. This is because the plant is self-pollinating, so the genotypes are the same. However, you will see problems where one parent might be part heterozygous, part homozygous, and the other might be all homozygous. There are many possibilities, so it's always the safe option to perform the distribution on both sides even if they are the same. So now that we have our dihybrid cross set up, we can start filling it out. We pretty much do the same thing as before with the Punnett squares. In the first box, we have big T, big Y, big Y. In the second, we have big T, big Y, little y. In the third, big T, big Y, big Y. And in the fourth, big T, big Y, little y. I'm now going to fill out the rest of the chart, so pause here to do it yourself and then press play when you're finished to compare. Now that we have finished the cross, we need to be able to quantify the phenotypic combinations. The possibilities are tall and yellow, tall and green, short and yellow, and short and green. Let's start with tall and yellow. Now, there should be the most squares with tall and yellow because those are both dominant phenotypes and therefore have higher probabilities of being expressed. As you can see here, 9 out of the 16 squares have tall and yellow pea plants. Next, we have tall and green. Because there are fewer combinations that make tall and green pea plants, there are only 3 out of 16. The same can be said for short and yellow plants, in which they also only have 3. Lastly, we have 1 pea plant out of 16 that is short and green due to both of these being recessive. This gives us an overall ratio of 9 to 3 to 3 to 1. Now, I have good news for you. Anytime you perform a dihybrid cross that is heterozygous across all genes, the ratio will always be 9 to 3 to 3 to 1. What the results of the dihybrid cross illustrated was that the two independently inherited traits gave us all of those combinations, therefore supporting the law of independent assortment. There are also 2 other types of genetic inheritance that do not necessarily abide by the laws of inheritance. Along with the typical dominant and recessive traits, we have incomplete dominance and codominance that we have to worry about as well. These forms of inheritance are called non-Mendelian genetics. First, let's start with incomplete dominance. This form of inheritance occurs when the offspring of a homozygous recessive parent and a homozygous dominant parent do not fully express either phenotype. Let's take these flowers for example. On the left, we have a red flower, and on the right, we have a white flower. Red flowers are dominant and white flowers are recessive. We cross a homozygous dominant red flower with a homozygous recessive white flower, and we get a heterozygous pink flower. Essentially, in incomplete dominance, a heterozygous offspring will produce a blended phenotype of the 2 alleles, in this case, pink flowers. The other type of inheritance is codominance. In this inheritance, 2 genes of the same trait can be dominant and both expressed equally in the offspring. An example of this is a white chicken and a black chicken are both dominant phenotypes, so when they mate, they produce a white chicken with black spots. Now, when we write out the Punnett square, rather than using the same letter as we have previously done, we will now write C to represent chicken, and in the top corner, we will write W for white and B for black. So the genotype of our white and black spotted chicken would be CBW. Another example of codominance that you may know of is our blood types. A and B are codominant and O is recessive. That is why someone could be blood type A, B, O, or AB. However, if someone that is homozygous A or homozygous B has an offspring with someone that is O, we can say with certainty that the offspring would be heterozygous with only the dominant A or B respectively being expressed. Codominance comes in when an A blood type and a B blood type have an offspring to create an AB blood type. If you know your blood type, consider asking your parents what theirs are so you can practice the Punnett score for yourself. And that's a wrap on Mendelian genetics, non-Mendelian genetics, and Punnett squares. Thanks for watching. In our next video, we will continue our discussion on genetic inheritance, but with an emphasis on pedigrees.